Have you ever paused after a meal, perhaps a hearty breakfast with eggs and bacon, or maybe a rich, creamy curry, and really considered the incredible invisible symphony playing out within your body to handle all that dietary fat?
Mm.
It's far more complex than just calories in, calories out, isn't
Oh,
it?
absolutely.
There's this biological ballet of breakdown, transport, and transformation happening at a microscopic level, all perfectly choreographed, that we rarely give a second thought.
Exactly. And, in a world that's just saturated with often conflicting health advice about fats, what you should embrace, what you need to avoid, it's really easy to feel lost.
Yeah, totally overwhelmed sometimes.
Right. But to truly cut through that noise, to understand why certain fats impact your health in specific ways or, why some individuals struggle with processing them,
Mm-hmm.
you really need to go beyond the headlines.
Mm-hmm.
You need a rock-solid, foundational understanding of how your body metabolizes every single lipid you consume.
And that, dear listener, is precisely our mission today on the Deep Dive. Welcome to a comprehensive, no stone unturned journey into dietary lipid metabolism.
Yeah, let's get into it.
Our goal isn't just to skim the surface, right? To give you a basic overview. Oh no, we're going deep. We want to immerse ourselves in every intricate detail, drawing directly from robust academic and research-based sources.
Mm-hmm.
We aim to equip you with the kind of foundational knowledge that, frankly, an elite professional in this field would possess.
That's the goal.
We're going to trace the path of lipids from the very moment they enter your body, following all their twists and turns, right through to their eventual use or storage. We'll be uncovering some surprising facts and truly intricate biological processes along the way. Get ready to master this information.
That's right. This isn't just about memorizing facts for the sake of it. It's really about understanding the why behind the what. We'll be delving into the biochemical pathways, the enzymes, the hormones, and these remarkable transport systems that collectively ensure your body can harness the power of fats. So, let's unlock this complex, yet I think incredibly elegant system together.
Okay, let's definitely unpack this. To truly master any subject, you really have to start with the fundamentals. So, first things first. What exactly are we talking about when we say lipids?
Excellent starting point. Yeah. Our sources immediately highlight that lipids are a remarkably heterogeneous group of organic molecules. You can think of them as a really diverse family, but they all share one defining, absolutely crucial characteristic: their water insolubility,
Okay.
meaning they are hydrophobic. And this single property, honestly, is the central tenet that underpins almost everything we'll discuss today about how the body has to handle them.
So, because they inherently repel water, they can't just dissolve and float freely in the watery environment of our cells or our blood. It's like oil and water, right? They sort of separate.
Precisely. That's the perfect analogy.
Mm.
Because of this inherent insolubility, body lipids are generally found in very specific, highly organized and compartmentalized ways.
Mm.
I mean, consider the lipid bilayer that forms the very structure of our cell membranes. It's a hydrophobic barrier, keeping the watery inside of a cell distinct from the watery outside, and actually within organelles too.
Right.
Or think about those tiny droplets of triacylglycerol, that's our primary storage fat, neatly tucked away inside adipocytes, our fat cells.
Mm.
And when these hydrophobic lipids do need to be transported through the blood, which is of course an aqueous solution, they just can't go it alone. They have to associate with specialized proteins. And this is where the concept of lipoprotein particles comes in, like tiny, sophisticated submarines for fat, you could say.
Mm.
Or even simply binding to larger proteins like albumin, which acts as a kind of general transport vehicle in the circulation for various hydrophobic compounds.
Right.
This fundamental challenge, how do you move water-hating molecules through water-loving environments? That's the stage upon which this entire intricate process unfolds.
That makes perfect sense. It really sets up this incredible biological engineering problem that the body has to solve. So when we talk about the different types of lipids, the sources mention triacylglycerols, cholesterol, phospholipids, and free fatty acids. Can you give us a quick snapshot of what these are and, how they typically appear in our diet?
Absolutely. Let's break it down. Think of triacylglycerols, often abbreviated as TAG, as the sort of big, bulky workhorses. They are by far the most abundant dietary lipid, making up well over 90% of our daily intake.
Wow, over 90%.
Yeah, it's the vast majority. Structurally, they're quite simple. Just 3 fatty acid chains esterified or linked to a single glycerol backbone. They're essentially the primary storage form of fat in our bodies and the main energy reservoir we consume.
So if I'm eating, say, a pat of butter or a spoonful of olive oil, that's predominantly TAG.
That's exactly right. Then we have cholesterol. Now, cholesterol is a type of steroid. It's found both free, meaning as the sterile molecule itself, and also in an esterified form called cholesterol esters, where a fatty acid is attached.
Okay.
Cholesterol gets a lot of press, sometimes bad press, but it's absolutely vital for cell membranes and also as a precursor for other important molecules like steroid hormones.
Right.
Then you have phospholipids. These are fascinating. They're similar in structure to TAGs, but instead of 3 fatty acids, they typically have 2, and then a phosphate group attached to that third position on the glycerol.
Ah, okay. The phosphate makes a difference.
Huge difference. That phosphate group gives them a hydrophilic or water-loving head and a hydrophobic water-hating tail. This dual nature makes them perfect for forming cell membranes, those bilayers we talked about.
Got it.
And finally, you have non-esterified or free fatty acids, often called FFA. These are just individual fatty acid chains not attached to a glycerol backbone. While they're a smaller portion of our diet, they are absolutely crucial building blocks. And our deep dive today will show how the body expertly processes all these diverse forms.
So we've established they're hydrophobic, and we've got our main cast of characters. But beyond just being a source of energy, which, let's face it, is what most people immediately think of when they hear fats, why do lipids truly matter? What other vital roles do they play?
That's a great question, because their roles extend far, far beyond just being fuel. While they are indeed a major energy source, providing a very dense and efficient form of stored energy for the body.
Yeah, more energy per gram than carbs or protein.
Yeah, exactly. Much more energy dense. But they also provide that absolutely crucial hydrophobic barrier we just mentioned. This barrier is the very foundation for the compartmentalization of the aqueous contents within our cells and all our subcellular structures. Without it, our cells wouldn't have their distinct boundaries. The cytoplasm wouldn't be separate from the extracellular fluid. And critically, organelles like mitochondria, the endoplasmic reticulum, the nucleus, they wouldn't be able to carry out their specialized functions in their own segregated, highly controlled environments. It's fundamental to the cell's architecture itself.
That's a powerful image. So they're not just fuel, they're literally the structural scaffolding, keeping everything organized and contained within our cells. What else? Because I know they're involved in signaling too, aren't they?
Precisely. Beyond structure and energy, lipids serve critical regulatory and coenzyme functions that basically orchestrate much of our body's daily operation. For instance, certain fat-soluble vitamins, that's A, D, E, and K, are, well, by definition, lipids. And they play absolutely vital roles in processes as diverse as vision, that's vitamin A, bone health and calcium regulation, vitamin D, antioxidant protection, vitamin E, and blood clotting, vitamin K.
Right, essential stuff.
Absolutely essential cofactors that the body often can't synthesize on its own in sufficient quantities. Furthermore, compounds like prostaglandins, which are derived from certain fatty acids, and steroid hormones, which are synthesized from cholesterol, play absolutely major roles in maintaining the body's homeostasis. They influence everything from inflammation responses and blood pressure regulation to metabolism and reproduction. These aren't just abstract biochemical roles. These are fundamental daily processes that keep your body functioning, adapting, and responding to its environment, often without you even realizing the lipid molecules working behind the scenes.
And this brings us, inevitably, to the flip side. What happens when this intricate system, this perfectly choreographed ballet, goes awry? Because imbalances in lipid metabolism can have truly profound and serious health consequences.
You're absolutely right. The sources really highlight that deficiencies or imbalances in lipid metabolism can lead to some of the major clinical problems physicians encounter globally.
Such as?
Well, we're talking about conditions like atherosclerosis, which is the progressive hardening and narrowing of arteries due to the buildup of plaque-containing lipids.
Yeah.
That's a primary underlying cause of heart attacks and strokes.
Okay.
We also see deep connections to diabetes, particularly type 2 diabetes, where lipid metabolism dysfunction often contributes significantly to insulin resistance. And of course, obesity, which is fundamentally a disorder of lipid storage and energy utilization. These aren't isolated health issues. They are broadly and deeply implicated in the epidemic of chronic diseases worldwide.
Yeah.
So understanding the fundamental processes we're about to delve into is not just academically interesting for someone wanting to be an elite professional. It's truly crucial for grasping the origins, progression, and potential therapeutic strategies for these widespread health challenges. This understanding provides the lens through which we can begin to see why things go wrong and maybe what might be done about it. It definitely foreshadows many of our later discussions on malabsorption and specific genetic disorders.
All right, now that we've set the stage with what lipids are and why they're so fundamentally important to pretty much every aspect of our health, let's actually embark on their extraordinary journey through the body. And that journey, perhaps surprisingly for some, actually begins, albeit in a limited way, right there in the stomach.
That's right, yeah. To put it in perspective, for most U.S. adults, the average daily intake of lipids is around 78 grams. That's quite a bit.
78 grams.
Yeah. And as we mentioned, over 90% of that substantial amount is triacylglycerol, or TAG. The remaining percentage is mainly made up of cholesterol, cholesterol esters, phospholipids, and a small but metabolically significant amount of non-esterified or free fatty acids.
Okay.
The key takeaway here is that these diverse forms all need to be meticulously broken down into absorbable units, and that initial breakdown for some of them starts earlier than many might assume, sort of preparing them for the intense processing ahead in the small intestine.
So digestion in the stomach is somewhat limited for lipids, but it's definitely not entirely absent. What specialized enzymes are at work there, navigating that, well, notoriously acidic environment?
Okay, so in the stomach, we encounter what are called acid lipases. This category includes lingual lipase, which, as its name suggests, originates from glands located at the back of the tongue. So digestion literally begins the moment food enters your mouth.
Wow, okay, from the tongue.
Yep. And then there's gastric lipase, secreted by the gastric mucosa, which is the lining of the stomach itself. Now, both of these enzymes are quite unique and remarkable because they're relatively acid stable.
Acid stable, meaning they like the acid.
Well, they tolerate it exceptionally well. Their optimal pH range is between 4 and 6, which is absolutely crucial given the highly acidic environment of the stomach, where the pH can plummet to 1.5 or 2. This stability allows them to function effectively where most other enzymes would simply be denatured and become useless.
And what's their specific job? Do they just indiscriminately break down all fats equally?
Not at all, and this is a really key detail. Their specific action is to hydrolyze, or you can say snip off, fatty acids from TAG molecules, but they exhibit a clear preference.
Okay.
They preferentially target those tags containing short or medium chain length fatty acids, meaning fatty acids with 12 or fewer carbon atoms in their chain.
Ah, the shorter ones.
Exactly. A classic and very important example of where these are found in abundance is milk fat, which contains a significant proportion of these shorter chain fatty acids. This preferential action is likely due to their active site architecture, which allows them better access to the ester bonds of these specific fatty acids, making them more accessible than the longer, bulkier chains found in most other dietary fats.
Ah, so that selective targeting makes them particularly important for, say, infants who are primarily consuming milk fat. This sounds like a brilliant biological adaptation.
Precisely. You've hit on a crucial aha moment there. For infants whose primary caloric source is often milk fat, these acid lithases play a critically important role in initiating the breakdown of those lipids.
Makes sense.
But their significance extends beyond infancy, illustrating a vital biological redundancy, a sort of backup system. Our sources highlight their critical importance for individuals with pancreatic insufficiency.
Okay, what's that?
That's a condition where the pancreas either produces insufficient digestive enzymes or where those enzymes just cannot reach the intestine. A prime example we'll touch on is cystic fibrosis.
Right.
In these cases, even with a near or complete absence of pancreatic lipase, the main fat-digesting enzyme, we'll discuss later, the really lingual and gastric lipases, can still degrade a significant portion of TAG molecules, especially those with short to medium chain fatty acids.
So they provide some digestion even when the main system is down.
Exactly. It means some level of lipid digestion can still occur, providing a crucial lifeline for nutrient absorption when the primary digestive system is compromised. It's a biological fallback mechanism, and frankly, it can be a lifesaver.
That brings us neatly to a crucial detour into cystic fibrosis, or CF. It's a condition often mentioned in the context of digestion, and our sources give us quite a detailed look at it, explaining why those stomach lipases become so vital.
Indeed. CF is a profoundly significant condition. It's actually the most common lethal genetic disease in Caucasians of Northern European ancestry, affecting approximately 1 in 3,300 live births in the United States.
Wow, that common?
Relatively, yes, for a genetic disorder. And it's an autosomal recessive disorder, which means an individual must inherit 2 copies of the mutated gene, one from each parent, to develop the condition. This highlights the importance of genetic counseling for carriers, of course.
And the genetic cause is related to something called the CFTR gene, right? I've definitely heard that acronym, but what does it actually do?
That's correct. The underlying genetic cause is specific mutations to the gene for the cystic fibrosis transmembrane conductance regulator, or CFTR, protein. Now, this protein is fascinating because it functions as a highly specialized chloride channel.
A channel, like a gate.
Exactly, a gate for chloride ions, located on the apical membrane, the surface facing the duct or lumen of epithelial cells. These are the cells that line ducts and passageways in various organs, including the pancreas, lungs, testes, and sweat glands. When the CFTR protein is defective due to these genetic mutations, it leads to a cascade of problems. Fundamentally, there's decreased secretion of chloride ions out of the cell, and as a consequence of that, there's increased reabsorption of sodium and water into the cell across these epithelial surfaces. This is the core cellular defect that drives essentially all the clinical manifestations of CF.
So how does that seemingly small cellular defect, just issues with a chloride channel, specifically snowball to impact lipid digestion and the pancreas so severely?
Right. It seems like a small thing, but the consequences are huge. The primary and most direct impact on the pancreas, and thus on lipid metabolism, is a profound change in the composition of pancreatic secretions. The depletion of water on the cell surface, because of that defective chloride transport and subsequent water reabsorption, results in the formation of abnormally thick, sticky mucus.
Ah, the thick mucus connection.
Exactly. This thick mucus then literally clogs the delicate pancreatic ducts, which are the conduits, the pipes, for digestive enzymes. When these ducts are clogged, it physically prevents the crucial pancreatic digestive enzymes, the real workhorses of digestion we'll talk about next, from reaching the small intestine.
So the enzymes are made, but they just can't get out.
Precisely. This leads directly to a severe condition known as pancreatic insufficiency. And this, as we noted just a moment ago, is exactly why those acid lipases in the stomach become so vitally important for these patients. They can perform some initial lipid digestion despite the profound pancreatic enzyme deficiency. It's a tragic example of how a single genetic defect can have such widespread and devastating consequences.
And beyond digestion, CF unfortunately has much broader impacts on the body, doesn't it? It affects way more than just the gut.
Yes, absolutely. While our deep dive today focuses specifically on lipid metabolism, it's crucial for anyone wanting to master this field to understand the systemic, multi-organ nature of CF.
Yep.
Beyond pancreatic insufficiency, CF also causes chronic lung infections with progressive pulmonary disease. That's often the most life-threatening aspect of the disease, leading to significant morbidity and mortality. It also typically leads to male infertility due to blockages in the reproductive tract and issues with sweat gland function, which is actually how the diagnostic sweat chloride test works. They measure the salt in sweat. The treatment for the digestive aspects typically includes aggressive replacement of the pancreatic enzymes that are missing or aren't reaching the intestine. They're administered as capsules with meals.
Enzyme replacement therapy.
Exactly. And additionally, supplementation of fat-soluble vitamins A, D, E, and K is critical, since their absorption is severely compromised due to the impaired lipid digestion and subsequent malabsorption. This truly illustrates how understanding these intricate digestive processes is fundamental for developing effective therapeutic interventions and improving quality of life for patients with conditions like CF.
So the stomach starts the process, especially for certain types of fats and in specific conditions like CF. But it definitely sounds like the real magic, or at least the bulk of the enzymatic breakdown, happens downstream in the small intestine. And before those powerful enzymes can do their heavy lifting, there's this crucial preparation step you mentioned,
Right.
emulsification.
Absolutely crucial, yes. Emulsification is a critical process that occurs primarily in the duodenum, which is the very first section of the small intestine, right after the stomach. Its primary purpose, its whole reason for being, is to dramatically increase the surface area of those hydrophobic lipid droplets.
Okay, why is surface area so key here?
Well, think about it like this. Imagine you have a large rock you need to dissolve. It would take ages, right? But if you crush that rock into a pile of fine sand, it dissolves almost instantly. Why? Because you've massively increased the surface area exposed to the solvent.
Right, more contact points.
Exactly. Lipids and enzymes are similar. If you have one large lipid droplet floating around, the digestive enzymes, which are water-soluble, can only work on its outer surface. But if you break that one large droplet into millions of tiny, stabilized microdroplets, you've massively increased the total surface area available for the enzymes to attack. Emulsification makes the enzymes work incredibly efficient, and therefore makes digestion possible on a meaningful scale within the time food spends in the gut.
That rock to sand analogy is perfect. It makes it really clear. So, how is this essential emulsification actually accomplished in the duodenum? What are the mechanisms at play?
There are two elegant and complementary mechanisms working in tandem, like a perfectly coordinated team. First, you have the powerful detergent properties of conjugated bile salts.
Okay, detergents, like soap.
Very much like biological soap, yes. And second, there's the mechanical mixing provided by peristalsis. Those are the rhythmic, wave-like muscular contractions of the intestinal wall that churn and mix the contents. Both are necessary. You need the chemical action and the physical action.
Let's delve deeper into these bile salts then. Where do they come from, and how do they function as these biological detergents?
Bile salts are truly fascinating molecules,
Yeah.
really master designers of the body's digestive chemistry. They are amphipathic derivatives of cholesterol.
Amphipathic, what does that mean?
It means they possess both a hydrophobic, lipid-soluble part, that's their bulky sterile ring structure derived from cholesterol, and a distinct hydrophilic, water-soluble part. They're meticulously synthesized in the liver from cholesterol, and then stored and concentrated in the gallbladder, ready for release into the duodenum, precisely when fats enter from the stomach.
Okay, and what gives them that hydrophilic part?
Chemically, a conjugated bile salt consists of that hydroxylated sterile ring structure, which is the hydrophobic core, with a side chain to which a molecule of either glycine or taurine-specific amino acids is covalently attached by an amide linkage. This glycine or taurine side chain provides the crucial hydrophilic water-loving portion of the molecule.
So, because of this dual nature, this sort of split personality, they can interact with both the fat and the water. How do these amphipathic bile salts actually perform their emulsifying magic with the fat droplets and the watery chyme in the duodenum?
Okay, so because of their dual nature, these emulsifying agents are perfectly designed to interact simultaneously with both the dietary lipid droplets and the aqueous duodenal contents. Imagine them surrounding the lipid droplets. Their hydrophobic parts burrow inward, burying themselves into the greasy core of the fat droplet,
Okay.
while their hydrophilic parts face outward, creating a water-friendly shell that interfaces with the aqueous environment. This coating does two crucial things. It stabilizes the smaller lipid droplets that are formed by the powerful mechanical churning action of peristalsis, and critically, it prevents them from coalescing or merging back together into larger, less digestible masses.
So they keep the tiny droplets tiny?
Exactly. They keep them dispersed and accessible. The source material mentions figure 15.3, showing glycolic acid, which really helps visualize this brilliant biological detergent system at work. It's an essential pre-treatment, setting the stage for the enzymes.
So once the lipids are emulsified and we have these tiny, stabilized droplets suspended in the watery chyme, then the real enzymatic breakdown by the pancreatic powerhouses can finally begin. This sounds like the main event of lipid digestion.
Exactly. This is where the majority, the sheer bulk of the enzymatic degradation of dietary triacylglycerols, cholesterol esters, and phospholipids actually occurs. These powerful enzymes are secreted by the pancreas, that gland sitting behind the stomach, and their secretion is tightly controlled by an elegant hormonal system, which we'll definitely get to shortly.
Okay.
Without this pancreatic arsenal, lipid digestion would be severely compromised, as we saw when we discussed cystic fibrosis.
Right. So let's start with the most abundant dietary lipid, triacylglycerol, or TAG. How does the pancreas unleash its power to break down these complex fats?
Okay, TAG molecules are simply too large, too bulky, to be efficiently absorbed directly by the mucosal cells lining the intestinal villi, those cells we call enterocytes. So they must be meticulously hydrolyzed or broken down into smaller absorbable units.
Makes sense.
The primary enzyme for this critical task is pancreatic lipase. It's a highly specialized esterase. It works by preferentially removing fatty acids from the first and third carbon positions, the outer positions of the glycerol backbone of the TAG molecule.
So it snips off the ones on the ends?
Precisely. The main products of this hydrolysis are a mixture of 2-monoacylglycerol, or 2-mag, that means a glycerol molecule, with just one fatty acid remaining, specifically at the second or middle carbon position, and free fatty acids, or FFA.
Okay, 2-mag and FFA.
Yep. And you can see this clearly illustrated in figure 15.2 in the source material, how those outer fatty acids are meticulously snipped off. This 2-mag, along with the FFA, is then readily absorbed by the enterocytes.
And how efficient is this pancreatic lipase? Is it easily overwhelmed if you eat a really fatty meal, or does the body produce it in abundance?
It's incredibly efficient. It's really a testament to the body's robust digestive capabilities. Pancreatic lipase is found in remarkably high concentrations in pancreatic secretions, making up about 2 to 3% of the total protein present in pancreatic juice.
Wow, that's a lot.
It is. And it has extremely high catalytic efficiency, meaning it can process a huge amount of substrate, the tags, very, very quickly. This high efficiency ensures that only severe pancreatic deficiency, like the kind we discussed in cystic fibrosis, where maybe 90% or more of pancreatic function is lost, results in significant malabsorption of fat, or steatorrhea. It's generally not a bottleneck unless there's a serious underlying condition.
But there's another protein involved, isn't there? Something that works in tandem with pancreatic lipase, cholipase. What's its role? Is it like a coenzyme or something else entirely?
Cholipase is truly the unsung hero here, a crucial facilitator for pancreatic lipase. It's also secreted by the pancreas, initially as an inactive precursor called procholipase,
Okay.
which is then activated in the intestine by another enzyme called trypsin. Once active, cholipase forms a very tight one-to-one binding complex with pancreatic lipase. Its crucial function is really twofold.
Okay, what does it do?
First, it acts as an anchor, physically binding the lipase enzyme to the lipid-aqueous interface right where the action needs to happen on the surface of those emulsified lipid droplets. This ensures the enzyme can interact effectively with its hydrophobic substrate.
Got it. Anchors it.
Right. And even more importantly, perhaps, it restores activity to pancreatic lipase even in the presence of inhibitory substances like bile salts.
Wait, bile salts inhibit the lipase, but I thought they were essential.
It's a bit ironic, isn't it? Bile salts are absolutely essential for emulsification, but if they completely coat the lipid droplet, they can actually interfere with the lipase's direct access to the tags inside. So cholipase acts like a crucial bridge, or maybe a molecular tether. It essentially displaces some bile salts at the interface and ensures the lipase can overcome this potential inhibition and do its job effectively, despite the challenging detergent-rich environment created by the bile salts. It's a really elegant solution to a complex biochemical problem.
That's truly clever molecular engineering by the body. Wow. And I think I've heard of a drug that actually targets these lipases, right, as a way to manage weight.
Yes, absolutely. A notable and clinically relevant example is orlistat. This is an anti-obesity drug that works precisely by inhibiting both gastric and pancreatic lipases.
How does that help with weight?
By inhibiting these enzymes, it directly prevents them from breaking down dietary fat, the tags, into absorbable units like 2-mag and FFA. The consequence is a significant decrease in the absorption of dietary fat.
So the fat just passes through.
Largely, yes. It leads to increased fat excretion in the feces, which in turn reduces calorie absorption from fat and can lead to weight loss. It's a direct pharmacological application of understanding this specific enzymatic process and its profound impact on nutrient absorption.
Right.
Of course, the potential side effects, like steatorrhea or fatty stools, provide a direct, albeit sometimes uncomfortable, demonstration of its mechanism of action. It shows the drug is blocking fat digestion.
Makes sense. Okay, so that covers the breakdown of the most abundant dietary lipids, the tags. What about cholesterol? Since some of that is also esterified in the diet, meaning it has a fatty acid attached, how is that handled?
Good question. While most dietary cholesterol is actually in its free, non-esterified form, about 10 to 15% is present as cholesterol esters.
Okay.
These esterified forms are hydrolyzed by another pancreatic enzyme called cholesterol ester hydrolase. It's also sometimes known as cholesterol esterase. This enzyme meticulously breaks them down into free cholesterol plus free fatty acids.
And does it need bile salts too?
Yes, interestingly, its activity is also greatly increased in the presence of bile salts. This again emphasizes the critical and multifaceted role of bile in overall lipid digestion. It's clearly not just for emulsification. It seems to optimize the function of several key enzymes.
And finally, the phospholipids. How are they degraded into absorbable units?
Right, the phospholipids. Pancreatic juice is also rich in the proenzyme, the inactive precursor of phospholipase A2. Like procholipase, this enzyme is activated by trypsin once it reaches the small intestine.
Activated by trypsin, okay.
And you guessed it, it also requires bile salts for optimal activity. Phospholipase A2 specifically removes one fatty acid, but it targets the fatty acid at the second carbon position of the phospholipid molecule.
Position 2 is specifically.
Yes. This action leaves behind a molecule called a lysphospholipid. For instance, phosphatidylcholine, which is the predominant phospholipid in our diet found in things like eggs and soy, becomes lysophosphatidylcholine after this initial step.
Okay, so one fatty acid off. What about the other one?
Good point. A subsequent enzyme, appropriately named lysophospholipase, can then remove the remaining fatty acid from the first carbon position. This leaves a glycerol-phosphoryl base, such as glycerol-phosphorylcholine. This base can then either be excreted in the feces, further degraded by other enzymes, or absorbed by the enterocytes. You can trace these meticulous transformations back to figure 15.2 in the source material to visualize the step-by-step products it shows: tags, cholesterol esters, and phospholipids all being broken down.
It's really clear that the pancreas is doing a huge amount of work here, a true chemical factory for digestion. How is all this enzyme secretion coordinated and regulated? It must be tightly controlled by the body to match the presence of food, right? You don't want enzymes flowing all the time.
It absolutely is, and this is a beautiful example of the body's intricate feedback mechanisms. Pancreatic secretion of these hydrolytic enzymes, which are so vital for degrading dietary lipids and proteins and carbs too, though we're focusing on lipids, in the small intestine, is tightly controlled by peptide hormones.
Hormones released where?
They're released from the intestinal mucosa itself, from specialized cells within the lining of the gut. It's an elegant system that ensures enzymes are delivered precisely when and where they're needed, basically, when food arrives.
And the sources mentioned two key peptide hormones involved here: cholecystokinin, or CCK, and secretin. Let's start with CCK. What triggers its release, and what are its critical actions?
Okay, cholecystokinin, or CCK, is a fascinating peptide hormone. It's produced by specialized endocrine cells called eye cells, which are located in the mucosa, the lining of the lower duodenum, and the jejunum, the first and middle parts of the small intestine.
And what makes them release CCK?
Its release is primarily triggered by the presence of lipids, specifically fatty acids and monoacylglycerols, the products of initial fat digestion, and also partially digested proteins entering these regions of the upper small intestine. These are the very nutrients that require the pancreatic enzymes and bile for further processing.
So the food itself triggers the signal.
Exactly. Once released into the bloodstream, CCK acts in a multi-pronged fashion, orchestrating several critical responses simultaneously.
Like what?
First, and critically for lipid digestion, it acts on the gallbladder, causing it to contract powerfully and release its stored bile, that vital mixture of bile salts, phospholipids, and free cholesterol, into the duodenum.
Okay, stimulates bile release.
Second, it potently stimulates the exocrine cells of the pancreas, prompting them to release their full complement of digestive enzymes: lipase, cholesterol ester hydrolase, phospholipase A2, and others.
Stimulates enzyme release. Got it.
And third, it also decreases gastric motility. That means it slows down the emptying of the stomach. This results in a slower, more controlled, and thus more manageable release of gastric contents, the chyme, into the small intestine. This provides ample time for the digestive and absorptive processes to occur efficiently, preventing an overload of the small intestine. You can see this beautiful hormonal orchestration visualized in figure 15.4 from the source.
And secretin. What's its unique role in this hormonal symphony, and what triggers its release? Is it also fats and proteins?
Secretin plays a different but equally crucial role. It's another important peptide hormone released by different intestinal cells, known as S cells, located primarily in the duodenum. Its release is specifically triggered not by the food components themselves, but by the low pH of the acidic chyme entering the intestine directly from the stomach.
Ah, so it responds to the acid.
Exactly. Secretin's primary and indispensable role is to cause the pancreas to release a voluminous solution rich in bicarbonate. This bicarbonate is essential because it acts as a powerful buffer.
To neutralize the acid.
Precisely. It neutralizes the acidic pH of the intestinal contents, bringing them rapidly up to the appropriate, more alkaline pH range, typically around pH 6 to 7, that is absolutely optimal for the activity of all those pancreatic digestive enzymes we just discussed.
Because those enzymes don't like acid like the stomach ones do.
It's exactly right. Without this rapid pH neutralization by bicarbonate triggered by secretin, those powerful pancreatic enzymes wouldn't be able to function effectively, even if CCK made the pancreas secrete them in abundance. They simply aren't stable or active at low pH. So secretin ensures the environment is absolutely perfect for pancreatic enzyme action. Together, CCK and secretin create the ideal conditions for digestion in the small intestine.
Okay, so digestion is largely complete now. Once these lipids are broken down into their smaller, more manageable components, the free fatty acids, the 2 monoacylglycerols, and the free cholesterol, what happens next? They're still technically outside the body, in the lumen of the small intestine. They have to actually get into the body, into your cells. This is the absorption phase, right?
Exactly. Now we move from digestion to absorption. The primary products of lipid digestion, those free fatty acids, especially the long-chain ones, free cholesterol, and 2-monoacylglycerol, 2-methyl-MAG, are absorbed predominantly in the jejunum, which is the long middle section of the small intestine. But here again, their hydrophobic nature presents a significant challenge in the watery environment of the intestinal lumen. They don't just spontaneously diffuse across the aqueous layer sitting right next to the cell membrane.
And this is where something called mixed micelles come into play, right? You mentioned bile salts help form these. Can you explain what these are, what they look like, and why they're so critical for the actual absorption process itself?
Yes, mixed micelles are truly ingenious molecular transporters, a masterclass in solubility solutions devised by the body. Imagine tiny disc-shaped clusters formed by these digestion products, the fatty acids, cholesterol, and 2-MAG, along with the bile salts that helped with emulsification in the first place, and also the essential fat-soluble vitamins, A, D, E, and K.
So they scoop everything up.
Pretty much all the lipid-soluble stuff, yes. They spontaneously self-assemble in a very specific ordered way right there in the intestinal lumen. Their hydrophobic roots, the greasy parts of the fatty acids, cholesterol, MAG, and the sterile part of the bile salts coalesce on the inside of the cluster. They form a greasy core that shelters this water-insoluble cargo.
Okay, hydrophobic core.
While their hydrophilic groups, primarily from the conjugated bile salts and maybe the head groups of any remaining lysophospholipins, face outward, this creates a water-friendly, soluble shell around the outside.
Ah, hydrophilic exterior.
Exactly. This structure makes the entire cluster soluble in the aqueous environment of the intestinal lumen. It effectively creates a tiny, sophisticated shuttle service for these otherwise insoluble lipids, allowing them to move through the water towards the cells. Figure 15.5 gives a wonderful visualization of these discoid or disc-shaped structures.
So they're like these microscopic transport bubbles, ferrying the lipids through the water right up to the absorptive surface of the intestinal cells. But how do they actually offload their cargo and get the lipids into the intestinal cells, the enterocytes?
Okay, so these mixed micelles diffuse through the lumen and approach the primary site of lipid absorption, which is the brush border membrane of the enterocytes. Remember, these are the intestinal mucosal cells that line the villi.
The ones with the finger-like projections.
Exactly, the microvilli, which dramatically increase the surface area for absorption. Now, importantly, there's a relatively still layer of water called the unstirred water layer, separating the bulk fluid of the intestinal lumen from the actual brush border membrane surface.
Okay, a water barrier.
Right. The hydrophilic surface of the micelles is crucial here. It allows them to efficiently transport these hydrophobic lipids through this unstirred water layer right to the membrane surface. Once at the membrane, the lipids, the FFA, 2-MAG, cholesterol, are released from the micelle and absorbed into the enterocyte. This happens primarily via diffusion down their concentration gradient, although some specific transporter proteins might also be involved, especially for cholesterol.
And the micelle itself, does it get absorbed too?
No, and this is absolutely critical to understand. The micelle itself is not absorbed. It merely acts as a delivery vehicle. It delivers its precious cargo to the membrane surface and then releases its components back into the lumen. The bile salts, now free again, are ready to diffuse back, pick up more lipids, form new micelles, and ferry them across. It's a continuous shuttle system.
That's a key distinction. The micelle is the shuttle, not the passenger.
Clever. And what happens to the bile salts after they've done their job forming these micelles and delivering their cargo multiple times? Are they just excreted from the body eventually? That seems wasteful. No, thankfully not. This is where the body's incredible efficiency truly shines again. The vast majority of the bile salts, we're talking well over 95%, are actively absorbed further down to the terminal ileum, which is the very last section of the small intestine, just before the large intestine.
Ah, they get reclaimed.
Exactly. They're reclaimed via a specific transporter system. This highly efficient recycling system is known as the enterohepatic circulation. It ensures that these valuable bile salts are returned via the portal vein back to the liver, where they can be taken up, maybe slightly modified, and then resecreted into the bile, ready to participate in subsequent rounds of lipid digestion and absorption.
Wow.
This recycling is absolutely vital for continuous lipid digestion, as synthesizing completely new bile salts from scratch using cholesterol is a complex and energy-intensive process for the liver. The body prefers to recycle.
You mentioned earlier that cholesterol is relatively poorly absorbed compared to other dietary lipids. Does the source elaborate on that, and are there clinical implications?
Yes, it's a significant point worth reiterating. The sources note that relative to other dietary lipids like fatty acids and MAG, cholesterol is indeed only poorly absorbed by the enterocytes. Typically only about 50% of the cholesterol that reaches the intestine. both from diet and from bile, is actually absorbed into the body.
Only half.
Yeah, which makes it a prime target for certain drug therapies aimed at lowering circulating cholesterol levels. For example, drugs like ezetimibe work specifically by inhibiting a cholesterol transporter protein called NPC1L1, located on the brush border membrane of the enterocytes.
So it blocks the gate for cholesterol?
Precisely. By doing so, it further reduces the absorption of both dietary and biliary cholesterol in the small intestine, providing a clinically effective pathway to lower plasma LDL cholesterol levels by directly blocking its entry into the body. It's a great example of applying this fundamental understanding of absorption mechanisms to therapeutic intervention.
And what about those short and medium chain fatty acids we talked about earlier, the ones that are important for infants and maybe CF patients? Do they also need mixed micelles for absorption, or do they have a different pathway altogether?
This is a fundamental distinction, really a point of true mastery in understanding lipid metabolism. Because short and medium chain fatty acids, SMCFAs, those with 12 carbons or fewer, are significantly more water-soluble than their long-chain counterparts.
Right, because they're shorter.
Exactly. They do not require the assistance of mixed micelles for their absorption by the intestinal mucosa. They can essentially bypass that entire micellar transport step.
So how do they get across the unstirred water layer?
They can diffuse directly through the unstirred water layer and across the brush border membrane into the enterocytes much more readily on their own. This direct absorption pathway is a complete game changer, setting them metabolically apart from the longer chain fatty acids that constitute the vast majority of our dietary fat intake. And this difference in absorption pathway becomes profoundly therapeutically significant, as we'll discuss shortly when we touch upon malabsorption conditions again.
Okay, so now we've got the lipid components, the long-chain free fatty acids, the 2-mag, and the free cholesterol inside the enterocytes, the intestinal cells. What happens then? Do they just stay as free fatty acids in 2-mag, or are they built back up into something else before they leave the cell to enter the rest of the body?
They're absolutely built back up. This is a critical step in the process. Once absorbed, these digestion products, the long-chain free fatty acids, free cholesterol, and 2-MG migrate within the cell to the smooth endoplasmic reticulum, or SER.
Okay, SER.
Okay, this organelle is the cellular hub where the intricate magic of biosynthesis of complex lipids takes place. The body is essentially taking the broken down parts it absorbed and rapidly rebuilding the larger, more complex lipid molecules it needs for transport out of the cell and into the circulation.
What's the very first step for those long-chain fatty acids once they're inside the enterocyte and heading to the SER? Do they need modification?
Yes, they do. For long-chain fatty acids, the very first step, a prerequisite for almost all subsequent reactions within the enterocyte, is their conversion into an activated form. This is an energy-requiring process.
Activated. How?
They are covalently attached to coenzyme A, forming a high-energy thioester bond. This creates fatty acyl coenzyme A, or simply fatty acyl-CoA. This activation step is catalyzed by an enzyme called fatty acyl-CoA synthetase, which is also sometimes known as thiokinase. This activation essentially primes the fatty acid, making it energetically ready for the complex synthetic reactions that follow in the SER. You can visualize this initial activation step, preparing the fatty acid for its next role in figure 15.6.
And once they're activated as fatty acyl-CoAs, how are the tags, phospholipids, and cholesterol esters, the complex lipids, actually resynthesized from these smaller components like 2-mag and free cholesterol?
Okay, using these newly activated fatty acyl-CoA derivatives, the 2-monoacylglycerol that was absorbed by the enterocytes is meticulously converted back into triacylglycerol, TAG, through a sequence of 2 rapid reacylations, basically adding 2 more fatty acids back onto the glycerol backbone.
Using specific enzymes?
Yes, this is carried out by 2 specific enzymes in the SER. First, acyl-CoA.monoacylglycerol acyltransferase, or MGAT, which adds a fatty acid to the first position, and then acyl-CoA.dietylglycerol acyltransferase, or DGAT, which adds the third fatty acid. This process efficiently rebuilds the primary storage and transport form of fat.
Okay, so tags are rebuilt. What about phospholipids and cholesterol?
Similarly, the lysophospholipids that might have been absorbed are reacylated using fatty acyl-CoAs to form intact phospholipids, ready to be incorporated into membranes or transport particles. This is done by a family of other acyltransferases, and the free cholesterol that was absorbed is also reesterified, predominantly by an enzyme called acyl-CoA.cholesterol acyltransferase, or ACA, to form cholesterol esters. The key insight here, the take-home message, is that almost all of the long-chain fatty acids entering the enterocytes are rapidly used in this fashion to reform TAG, phospholipids, and cholesterol esters. They don't just hang around as free fatty acids inside the cell, they are immediately repackaged for transport out.
So it's essentially a rapid reassembly line inside the enterocyte, taking those broken down parts absorbed from the gut lumen and quickly rebuilding the larger lipid molecules needed for transport. Is this the fate for all absorbed fatty acids, or do those unique short and medium chain fatty acids have a different internal fate too inside the enterocyte?
You're astute to keep tracking those. This is where the unique path of the short and medium chain fatty acids becomes even more pronounced, really differentiating them metabolically from the long chain ones. No, short and medium chain fatty acids, SMCFAs, are not converted to their CoA derivatives within the enterocyte.
They skip the activation step.
They skip the activation step, and consequently, they are not reesterified to 2-meg to form tags inside the enterocyte. This is a crucial difference.
So what do they do inside the cell?
Instead, once inside the cell, they are directly released from the enterocyte into the portal circulation.
The portal vein, straight to the liver.
Exactly. They go straight from the intestinal cells through the portal vein directly to the liver. They don't enter the lymphatic system like the long-chain fats do. They carried in the portal blood, primarily associated with serum albumin, that transport protein we mentioned.
Wow, completely different route.
Completely different. This dramatically different metabolic fate, bypassing intracellular reesterification and bypassing the entire lymphatic system, is precisely why they're so important in certain clinical scenarios where the conventional long-chain lipid absorption pathways are compromised.
And speaking of clinical scenarios, what happens when this entire absorption and resynthesis process fails or is severely impaired? You mentioned steatorrhea before.
Right. When lipid malabsorption occurs, for whatever reason, it results in that clinical condition known as steatorrhea. This is essentially the presence of increased lipid undigested or unabsorbed fat, often visible as greasy, foul-smelling, bulky stools in the feces.
Okay.
It's a clear and unmistakable sign that the body is not absorbing dietary fats properly. And importantly, this also means that the fat-soluble vitamins, A, D, E, K, and essential fatty acids, which rely on the same absorption pathways, micelles, etc., are also likely being malabsorbed, leading to potential deficiencies of those crucial nutrients as well. You can visualize the stark consequences in figure 15.7 from the source material.
And you've already mentioned a couple of conditions earlier that could lead to this kind of malabsorption. Can you just bring those back into context quickly?
Exactly. We've already discussed how cystic fibrosis can cause steatorrhea, but in that case, remember, it's primarily due to poor digestion of lipids, because the pancreatic enzymes can't reach the intestine to break them down in the first place.
Right, digestion failure.
Another major condition that leads to steatorrhea is short bowel syndrome. Here, perhaps due to surgical removal of a significant portion of the small intestine, maybe because of Crohn's disease or injury, or severe damage to the intestinal lining, there's simply not enough functional absorptive surface area.
So absorption failure.
Exactly. So these illustrate different origins, a problem with digestion versus a problem with absorption, both potentially leading to the same clinical outcome of steatorrhea. Understanding the specific root cause is absolutely crucial for tailoring effective treatment.
And this brings us back full circle to the practical importance of those short and medium chain fatty acids in a clinical setting. How do they fit into the therapeutic picture for conditions causing steatorrhea?
Absolutely. The unique ability of short and medium chain fatty acids, SMCFAs, to be taken up by enterocytes without the aid of mixed micelles, and then to directly enter the portal circulation, bypassing the need for reesterification and chylomicron formation. Well, that has made them incredibly important in medical nutrition therapy for individuals with various malabsorption disorders.
How are they used?
Products like MCT oil, which stands for medium-chain triglyceride oil, are rich in these SMCFAs. They are a vital nutritional tool. Because these fats don't require the compromised digestive pathways, like pancreatic lipase, or the complex absorptive pathways, like micelle formation and chylomicron packaging, that long-chain fatty acids do, they can serve as a vital, readily absorbable source of calories and some essential fatty acids for these patients. This helps to mitigate the severe nutritional deficiencies and weight loss often associated with chronic steatorrhea. It's a direct, sometimes life-changing application of understanding these distinct metabolic pathways.
Okay, so inside the enterocytes, the long-chain fatty acids have been reassembled into complex lipids, primarily tags, but also cholesterol esters and phospholipids. We know they're highly hydrophobic. They can't just float freely out of the cell and into the bloodstream to be delivered to the rest of the body. How does the body package and transport these newly synthesized lipids to where they need to go? This sounds like another major transport challenge.
You've hit on the next grand challenge for the body. Absolutely. They are indeed very hydrophobic and would simply aggregate into large, insoluble clumps within the aqueous environment of the cell, or certainly in the lymph or blood, if they weren't properly handled.
Right, like oil droplets merging.
Exactly. Therefore, they must be meticulously packaged into specialized lipoprotein particles before they leave the enterocyte. This packaging is absolutely essential because it does two things. It stabilizes the large lipid droplet core, and it dramatically increases its solubility and dispersibility in an aqueous environment. This prevents multiple particles from coalescing into larger problematic masses that could potentially clog up the circulation. It's like putting that oil-filled balloon inside a water-friendly wrapper, as he said earlier.
And what does this elaborate package actually consist of? What are these specialized lipoprotein particles called, the ones made in the intestine?
This specific package assembled in the enterocyte to transport dietary fat is called a chylomicron. It's really a marvel of biological engineering. A chylomicron is essentially a large, spherical lipid droplet at its core, primarily composed of those newly resynthesized triacylglycerols, tags, and also the cholesterol esters.
Okay, the greasy core.
Right, and this core is surrounded by a very thin outer layer or shell. This outer layer, acting as the wrapper, is composed of phospholipids, with their hydrophilic heads facing out, some non-esterified cholesterol interspersed within the phospholipids, and crucially, a large structural protein molecule called apolipoprotein B48, or ApoB48.
ApoB48, that's specific to chylomicrons.
Yes, ApoB48 is unique to chylomicrons produced by the intestine. This entire envelope, phospholipids, free cholesterol, and ApoB48 acts as the stable interface, ensuring the hydrophobic lipid core remains perfectly stable and soluble, or at least dispersible, in the watery environment of the lymph and blood.
And is there a specific protein that helps with the intricate assembly of these chylomicron particles inside the cell? Putting the lipids and the ApoB48 together sounds complicated.
Yes, there absolutely is, and it's indispensable for the process. A protein called microsomal triglyceride transfer protein, or MTP, is absolutely essential for the proper assembly of all triacylglycerol-rich, ApoB-containing lipoprotein particles. That includes not just chylomicrons made in the intestine, but also VLDL particles made in the liver, which we might discuss another time.
Okay, MTQ. What does it do?
It functions within the endoplasmic reticulum, the SCR, facilitating the loading of the large core of TAG onto the growing ApoB48 molecule as it's being synthesized. Without functional MTP, these chylomicrons simply cannot be properly formed or secreted from the enterocyte. Mutations in the MTP gene lead to a rare but severe malabsorption disorder called abitopliproteinemia, where these particles can't be made.
Wow. So these fully assembled chylomicrons are packed with dietary fat inside the enterocytes. How do they exit the cells, and what's their first destination for transport within the body? Because you mentioned earlier it's not directly into the bloodstream, is it?
That's a key distinction, and it goes back to their sheer size. Chylomicrons are actually enormous by cellular standards, far too large to squeeze into the tiny blood capillaries directly adjacent to the enterocytes.
Okay, too big for blood vessels initially.
Right. So they released from the basolateral side of the enterocytes, the side facing away from the gut lumen, by a process called exocytosis, where the cell essentially expels them and closes in a vesicle that fuses with the cell membrane. Their first destination is not the bloodstream, but rather the lacteals.
Lacteals, what are those?
These are specialized, blind-ended lymphatic vessels located right within the core of the villi of the small intestine. The lymphatic system has larger pores than blood capillaries, allowing these big chylomicron particles to enter.
Ah, the lymph system first.
Exactly. And the presence of these large fat-laden particles in the lymph after a lipid-rich meal is actually quite visible. It gives the lymph a characteristic milky or creamy white appearance. And this milky lymph has a specific name.
Which is chyle, right. Not to be confused with chyme, you mentioned clarifying this earlier.
Absolutely. It's a great aha moment for many learners and crucial for professional clarity. This lipid-rich lymph, collected from the intestine containing the chylomicrons, is specifically called chyle. Chyle. It's vital to distinguish it from chyme, Chyme, which is the semi-fluid mass of partially digested food that passes from the stomach into the duodenum.
Got it. Chyme is stomach stuff, chyle is fatty lymph.
Perfect. And the particles themselves floating within the chyle are named chylomicrons. Using these terms precisely is definitely key for mastering this topic.
Perfect. That's crystal clear now.
Yeah.
So where do these chylomicrons go from the lymphatic system, from the lacteals? What's their rate to eventually reach the general circulation, the bloodstream?
From the lacteals within the intestinal villi, these chylomicrons travel through progressively larger lymphatic vessels. They merge and flow upwards until they reach the thoracic duct, which is the largest lymphatic vessel in the body, running up through the chest.
Okay, the main lymph highway.
Pretty much. From the thoracic duct, the chyle containing the chylomicrons is finally conveyed into the bloodstream, where the thoracic duct empties into the left subclavian vein near the collarbone, so they enter the general blood circulation there.
Why the detour through the lymph? Why not straight to the liver via the portal vein, like the short-chain fatty acids?
That's a great question. This indirect route via the lymph system, bypassing the liver initially, is thought to serve a couple of purposes. It prevents the liver from being suddenly overwhelmed by a massive influx of dietary fat directly after a meal. It allows the fat to be distributed more gradually to peripheral tissues first, and, as we noted, the chylomicrons are likely just too large to enter the portal capillaries directly. Figure 15.6 in the source material nicely summarizes these intricate steps in their production, secretion into lymph, and eventual entry into the blood.
Okay, so they finally make it into the bloodstream. Are they fully functional and ready to deliver their lipid cargo, the tags, when they first enter the blood? Or do they need to mature somehow, perhaps pick up additional components?
Another excellent question, hitting on a key point. They actually need to mature once they enter the blood. When they're first released from the lymph into the blood, they are considered nascent or immature chylomicrons.
Nascent, okay. What makes them mature?
To become fully mature in functional meaning, ready to interact with the enzymes and receptors that will ultimately strip away their lipid cargo and allow them to be cleared, they need to acquire additional apolipoproteins. Specifically, they pick up apolipoprotein E, ApoE, and apolipoprotein C2, ApoC2.
Where do they get those from?
They acquire them from high-density lipoproteins, or HDL, which are other smaller lipoprotein particles already circulating abundantly in the blood. HDL essentially acts as a circulating reservoir for these exchangeable apolipoproteins.
And why are ApoE and ApoC2 so important for the chylomicron?
They're absolutely crucial. ApoC2 acts as an essential activator for the enzyme lipoprotein lipase, LPL, which is the enzyme that will break down the tags in the chylomicron. ApoE serves as the ligand, the key, recognized by effectors, particularly on the liver, that are responsible for clearing the chylomicron remnants from the circulation later on. Without acquiring these from HDL, the chylomicron can't effectively deliver its fat or be properly cleared.
So now we finally have these mature chylomicrons circulating in the blood. They're essentially huge lipid delivery trucks loaded mainly with triacylglycerols and some cholesterol esters. How do the peripheral tissues that need this fat, like our muscles and our fat cells, actually get access to these lipids? How is the cargo unloaded from the circulating trucks?
Right, the delivery phase. Once in the bloodstream and fully matured, the vast majority of the triacylglycerol, TAG, contained within these chylomicrons is efficiently broken down and delivered right where it's needed, in the capillary beds. These are the tiniest blood vessels that permeate our metabolically active tissues, primarily our skeletal muscle for movement, cardiac muscle, our heart is constantly working and loves fatty acids, and adipose tissue, which are our main fat storage sites. These tissues have a high demand for fatty acids, either for immediate energy production or for storage for later use.
And what's the specific enzyme responsible for this crucial breakdown of TAG in the capillaries, the enzyme that allows the tissues to actually take up the fatty acids from the chylomicron?
The key enzyme here, the molecular scissors that perform the unloading of the cargo, is lipoprotein lipase, or LPL. We just mentioned its activator, ApoC2. LPL is responsible for rapidly hydrolyzing or degrading the triacylglycerol core within the chylomicrons.
What does it break TAG down into?
It breaks TAG down into free fatty acids, FFA, and glycerol. This enzymatic action occurs right at the capillary wall, making the released free fatty acids immediately available for uptake by the adjacent tissue cells, the muscle cells or the fat cells.
Where exactly is LPL located? How does it manage to access the TAG deep inside the chylomicrons, which are circulating within the blood vessels? It sounds like a very precise placement is needed for it to work.
LPL is quite cleverly positioned, really a masterstroke of biological design for efficient nutrient delivery. It's synthesized and secreted primarily by the cells that will use the fatty acids, namely adipocytes, our fat cells, and muscle cells, both skeletal and cardiac.
Okay, made by those cells, but where does it sit?
Once secreted from these cells, it doesn't just float freely in the tissue fluid or the blood. Instead, it's transported to and anchored to the luminal surface, the inner lining facing the blood of the endothelial cells that form the walls of the capillaries within those very same muscle and adipose tissues.
Ah, so it's stuck to the inside wall of the blood vessel.
Exactly. It's attached via interactions with heparan sulfate proteoglycans on the endothelial cell surface. This strategic positioning means LPL is right there, at the interface between the circulating chylomicrons flowing past in the blood and the tissue cells just outside the capillary wall. It's perfectly positioned to interact with the chylomicrons activated by ApoC time and strip away their tag cargo as they pass by. It's like having a dedicated unloading dock right at the tissue's doorstep.
That's incredibly efficient. So what happens if there's a problem with LPL itself, maybe a genetic defect, or with its essential coenzyme ApoC time? Are there clinical consequences if this crucial unloading step fails?
Absolutely, and the consequences can be quite severe, illustrating just how vital this enzyme is. The sources highlight a rare but very significant genetic condition called familial chylomicronemia, which is also known as type hyperlipoproteinemia.
Okay, what causes it?
It's an autosomal recessive disorder, meaning it's inherited, caused by a deficiency, either a complete lack or severely reduced function in either LPL itself or its essential coenzyme, apolipoprotein C2. Yeah, C2. If either part of that system is broken, the result is the same.
And what is the result?
The profound result is that chylomicrons cannot be efficiently cleared of their tag cargo. This leads to a persistent and dramatic presence of chylomicrons in the blood, even after an overnight fast a condition known as fasting chylomicronemia. Normally, chylomicrons should be cleared within hours after a meal. This leads to severe hypertriacylglycerolemia, meaning extremely high levels of triacylglycerols in the blood, often thousands of mg/dl when normal is under 150. The blood plasma can literally look milky or creamy because of all the fat.
Wow. And are there health problems associated with that?
Yes, serious ones. This massive accumulation of chylomicrons can lead to recurrent episodes of acute pancreatitis, which is a very painful and potentially life-threatening inflammation of the pancreas. Patients may also develop eruptive xanthomas, small, yellowish, fat-filled bumps that appear suddenly on the skin, especially over pressure points or buttocks. This condition really underscores just how absolutely crucial LPL activity is for the proper and safe metabolism of dietary lipids and preventing dangerous accumulation.
Okay, now let's assume LPL is working correctly. Once it has done its job, breaking down the tag in the chylomicron, what happens to the products, the free fatty acids, the glycerol, and whatever is left of the chylomicron particle itself? Let's start with the free fatty acids that were just released. What are their fates?
Right, so the free fatty acids, FFA, derived from the hydrolysis of TAG by LPL, have a couple of primary fates, really depending on the needs of the tissue and the body's overall energy status at that moment. They can either be directly taken up by the adjacent muscle cells and adipocytes, crossing the capillary endothelium and the cell membrane, where they are then immediately available for use within those cells. Or, if they're not immediately taken up by the local cells, perhaps because supply exceeds immediate demand, they can enter the general circulation. In the blood, they don't travel freely because they're still hydrophobic. They associate with serum albumin.
Albumin, again, the transporter protein.
Exactly. Albumin is a large, very abundant protein secreted by the liver that acts as a general transporter for a variety of hydrophobic compounds in the blood, including these free fatty acids, bilirubin, and even some drugs. Albumin carries them safely through the bloodstream to other tissues that might need them.
And once inside the cells, what do the cells do with the fatty acids?
Once inside the cells, most cells throughout the body, muscle, liver, heart, etc., have the capacity to oxidize these fatty acids through a process called beta oxidation to produce a large amount of ATP, or energy. Fatty acids are a major fuel source, especially during fasting or prolonged exercise.
Right, fuel. What about storage?
Adipocytes, our fat storage cells, have a crucial additional role. They can rapidly take up these free fatty acids and reesterify them, link them back onto a glycerol backbone to synthesize new triacylglycerol molecules. These tags are then stored very efficiently as large lipid droplets within the fat cell. This stored fat serves as the body's main energy reserve, ready to be mobilized and released back into the circulation as FFA when energy is needed later, perhaps during periods between meals or overnight fasting.
Okay, so fatty acids are used for energy or stored as TAG. What about the glycerol backbone that's released from the TAG when LPL breaks it down? Does it follow the same path as the fatty acids, or does it have a different destination?
No, glycerol has a distinctly different and unique path. Unlike the fatty acids, glycerol released from TAG hydrolysis by LPL is highly water-soluble.
Oh,
It doesn't need albumin to travel in the blood. It's primarily taken up from the blood by the liver. The liver is unique, or at least highly specialized, in its ability to metabolize glycerol. Most other tissues, like muscle and adipose tissue, lack the key enzyme.
What enzyme is that?
Once inside the hepatic cells, liver cells, glycerol is phosphorylated by a specialized enzyme called hepatic glycerol kinase to produce glycerol 3-phosphate.
Glycerol 3-phosphate. What can the liver do with that?
This glycerol 3-phosphate is actually quite versatile and acts as an important intermediate, an entry point into several major metabolic pathways within the liver. It can enter either glycolysis, the pathway for glucose breakdown to produce energy, or gluconeogenesis, the pathway for synthesizing new glucose, especially during fasting. It enters these pathways via its conversion to dihydroxyacetone phosphate, DHAP, which is an intermediate in both.
Okay, energy or glucose production. Anything else?
Yes, the glycerol 3-phosphate can also be directly used by the liver as the backbone for the synthesis of new triacylglycerols within the liver itself. These liver-synthesized tags can then be stored briefly in the liver or packaged into other lipoprotein particles, like VLDL, for export to other tissues. So the glycerol is fully recycled and efficiently integrated back into the body's central metabolism. It's not wasted.
So after the chylomicrons have circulated and LPL has stripped away most of their tag cargo, delivering the fatty acids to the peripheral tissues, what's left of the original particle, and what happens to these remaining chylomicron remnants? They can't just keep circulating forever, can they?
No, they definitely can't. After the vast majority of the triacylglycerol has been removed by the action of LPL in the capillaries of muscle and adipose tissue, what's left are particles called chylomicron remnants.
What are they like compared to the originals?
These remnants are significantly smaller and denser than the original nascent chylomicrons because they've lost most of their bulky, buoyant tag core. However, they still contain significant amounts of the cholesterol esters that were absorbed from the diet or packaged in the intestine, along with the phospholipids from the original shell, some of the apolipoproteins, importantly they retain ApoE but may lose some ApoCII, all the fat-soluble vitamins, A, D, E, K, Sh, that were originally absorbed with the dietary fats, and usually a small residual amount of triacylglycerol that LPL didn't remove.
So they're relatively enriched in cholesterol now. Where do they go?
These remnants, now relatively rich in cholesterol compared to TAG, continue to circulate briefly, but then travel through the bloodstream to the liver. The liver is the primary site for their clearance from the circulation.
How does the liver grab them?
They bind to specific receptors on the surface of liver cells, hepatocytes. The key interaction involves apolipoprotein E, ApoE, on the surface of the remnant acting as the crucial ligand, essentially the key that fits into specific liver receptors, like the LDL receptor or the LDL receptor-related protein, LRP.
ApoE is the key. Okay.
Once bound to these receptors, the entire chylomicron remnant particle is taken into the liver cells through a process called receptor-mediated endocytosis, where the liver cell membrane essentially engulfs the remnant particle, bringing it inside in a vesicle.
And what's inside the liver cell?
Inside the liver cells, these internalized remnants fuse with lysosomes, which contain powerful hydrolytic enzymes. These enzymes then completely break down the remnant particle back into its constituent components, free cholesterol, fatty acids, amino acids from the apolipoproteins, glycerol, phosphate, etc.
And can the liver reuse those parts?
Absolutely. The valuable components, such as the cholesterol and the nitrogenous bases from phospholipids, like choline, can then be recycled and reused by the liver for various purposes, like synthesizing new lipoproteins, like VLDL, synthesizing bile acids, or incorporating into membranes. The liver is the body's central processing and recycling hub for lipids.
And if this crucial chylomicron remnant removal process by the liver doesn't work correctly, if the liver can't clear these remnants efficiently, are there clinical consequences similar to the LPL deficiency we discussed earlier?
Absolutely. If the removal of these chylomicron remnants by the liver is significantly decreased, this leads to their accumulation in the plasma. This often happens due to impaired binding to their specific liver receptors. A common cause is a genetic defect in ApoE itself, particularly inheriting two copies of a variant called ApoE2, the ApoE2E2 genotype, which binds poorly to the liver receptors.
Okay, faulty ApoE.
Exactly.
Yeah.
This leads to a condition called type 3 hyperlipoproteinemia, which is also known as familial dyspeptidolipoproteinemia or sometimes broad beta disease because of how the lipoproteins look on electrophoresis.
What does that cause?
The accumulation of these remnants in the plasma leads to significantly elevated levels of both cholesterol and triacylglycerols, because the remnants contain substantial amounts of both. Clinically, this can manifest as premature atherosclerosis, leading to early heart disease and peripheral vascular disease, and characteristic fatty deposits called xanthomas, particularly tuberous xanthomas, larger, softer deposits often on elbows and knees, and distinct palmar xanthomas, yellowish deposits that appear in the creases of the palms. This condition powerfully demonstrates the critical importance of this final remnant clearance step in chylomicron metabolism for maintaining healthy lipid levels and preventing cardiovascular pathology.
Wow, that was truly a deep dive, wasn't it? A complete mastering of the journey, really. We started with the basic definition of lipids, hydrophobic molecules, and ended all the way with how their remnants are meticulously cleared from the bloodstream by the liver.
It has been an extensive journey, hasn't it? We've really traced dietary lipids from the moment of their ingestion, through their initial limited breakdown in the stomach by those crucial acid lipases, remember, a critical fallback for infants in conditions like cystic fibrosis, then onto their essential emulsification in the small intestine, brilliantly aided by bile salts, acting as detergents, and the mechanical churning of peristalsis. We saw their extensive enzymatic degradation by those pancreatic powerhouses like pancreatic lipase, with its helper cholipase, cholesterol ester hydrolase, and phospholipase A2.
Yeah, and the hormones. We explored the crucial hormonal orchestration by CCK, bringing out the bile and enzymes, and secretin, neutralizing the acid to make the environment just right.
Exactly. Then we followed their fascinating absorption into the enterocytes, often via those clever mixed micelles acting as shuttles. And crucially, inside the cells, they're not just absorbed, but meticulously resynthesized into complex lipids like tags, phospholipids, and cholesterol esters.
And we definitely unpacked the unique and clinically significant pathways of the short and medium-chain fatty acids compared to their long-chain counterparts. Remember, bypassing micelles, bypassing reesterification, going straight to the portal circulation via albumin. That's such a key insight for therapeutic nutrition.
Absolutely. And we didn't just stop at absorption, did we? We then delved into the sophisticated packaging of these newly reformed long-chain lipids into chylomicrons within the enterocytes, involving ApoB48 and MTP.
Right.
We clarified their strategic release into the lymphatic system as Kyle mastering that term, distinguishing it from chyme.
Right.
We followed their entry into the bloodstream via the thoracic duct, their essential maturation by acquiring ApoE and apoC from HDL, and finally, their targeted delivery of tags to peripheral tissues via the indispensable lipoprotein lipase, anchored on the capillary walls.
And to complete the entire cycle, we dissected the ultimate fates of the products, the free fatty acids going for energy or storage, the glycerol heading uniquely to the liver for recycling, and the meticulous clearance of the cholesterol-enriched chylomicron remnants returning to the liver, recognized by ApoE for final processing and recycling.
Phew. It's a lot when you lay it all out.
It really is. And throughout this entire intricate narrative, we've seen time and again how understanding these minute interconnected biochemical pathways provides such a powerful foundation for appreciating both profound health and complex disease states. Conditions like cystic fibrosis, the debilitating effects of steatorrhea, the severe implications of familial chylomicronemia due to LPL or apoC deficiency, and the challenges of type 3 hyperlipoproteinemia from poor remnant clearance. They all become far more understandable, less like random events when you grasp the underlying molecular ballet. It's truly incredible how perfectly interconnected all these processes are, and the crucial, often unsung roles played by various enzymes, hormones, transport molecules like bile salts and albumin, and structural proteins like the apolipoproteins.
And I think that distinction between the short medium-chain fatty acids and the long-chain fatty acids and their fundamentally different metabolic fates right from absorption through to transport truly stands out as a recurring theme and a key insight. It bridges normal physiology or critical therapeutic approaches in a really clear way. Mastering this depth really allows you to see the elegance and the incredible precision of the human body's biochemistry.
So the next time you enjoy a meal containing fats, whether it's a piece of fatty fish like salmon, some nuts, avocado, or even just a simple dressing on your salad, maybe take just a moment. Consider the silent, incredibly complex, and perfectly choreographed ballet that is happening within your body right then to break it down, transport it, and put it to work with such astounding precision. What new questions does this deep dive spark in you about your own body's amazing hidden chemistry?